you so much, Liz, for the very kind introduction, and thanks to all of you for coming today. I'm really excited to tell you a little bit about the work that I've been doing to untangle the complicated evolutionary history of convergent color pattern in New World snakes, and how their example may serve as a window into understanding both pattern and process and evolutionary convergence more generally. Now, to set the stage, I want to explain that this is more than just a pretty title slide. So the two clades of snakes that are represented here, this is a venomous elapid coral snake on your right and a harmless colubrid snake on your left. These two clades of snakes diverged from each other just after the end of the Cretaceous, about 65 million years ago, right about the same time as the major crown diversification of most orders of modern mammals. So these are two very, very different snakes. The individuals that are depicted here are illustrated from two real museum specimens. These two individuals were collected together at the same field site on a UMMZ expedition to Guatemala in 1940. And yet, despite the great length of time since their most recent common ancestor and the fact that the vast majority of snakes in the world are some variation of brown, these two species now coexist together in space and time in almost perfect replicas of the same very conspicuous color pattern. So today I'm going to tell you a little bit about how and why we think that came to be, which does come with some surprises. Perhaps more excitingly, how likely such a thing is to be true millions of years from today. And so to help you to have some context for understanding why I've pursued research on mimetic convergence, I want to say a few brief words about the major themes of my overall research program. So my research interests span a broad range of topics across both ecology and evolution, as well as some applied conservation biology. I've worked on a number of different systems and a number of different questions over the course of my research career so far. However, one common characteristic that unites most of the research that I do is asking big picture questions about trait evolution, especially about the ecological factors that drive the origin and stability of novel traits over time. Now, I study all kinds of traits, which often co-vary, so I tend to rely pretty heavily on interdisciplinary approaches to the questions that I ask. Now, I spent most of my early career investigating the origin of cooperative nuclear family social groups in lizards, in which I described an independent origin of complex sociality in vertebrates. Now here you see one of the species that I studied. This is the desert night lizard that inhabits the Joshua Tree woodlands of California. And I combined experimental manipulations in both the lab and the field with molecular analyses of relatedness to look at not only the mechanisms but also the fitness consequences of the transition between solitary and social behavior. Why become social? I also spent about 4 years investigating variation in mating systems and the role that alternative mating strategies can play in promoting reproductive isolation. So here you can see 6 polymorphic throat color phenotypes found in a single population of lizards. And these phenotypes correspond to 3 different mating strategies that cycle over time through rock, paper, scissors dynamics, in which each strategy can beat and is beaten by one of the other strategies in the system. Now, I investigated the reproductive consequences of shifts between a polymorphic system and a monomorphic system in 2 independent origins of rock, paper, scissors dynamics, in the side-blotched lizard in the US and Mexico, and in the common lizard in Europe. So now, all of these projects, whether they're on sociality or mating systems or mimicry, they're all fundamentally driven by my desire to understand how and why we see shifts to new and sometimes very distinct states in trait space, and especially how reversible these transitions are over both ecological and evolutionary timescales. And this focus has led to my appreciation for the power of evolutionary convergence, multiple independent origins of the same trait, for not just characterizing species-specific patterns of change, but for actually having statistical power, replicates, in order to test hypotheses about trait evolution in nature, which is what I'm going to talk about today. So my talk will be divided into 2 main parts. First, I'm going to give some background about one of the ultimate examples of convergence, and that's mimicry. A little bit about what we know and what we predict about how mimicry should operate, and how we can use snake mimicry to test hypotheses about evolutionary trajectories of traits across all of New World snakes. Then we're going to look at a single clade in depth, which allows us to follow the mimetic phenotype over the landscape of both time and space, and test among mechanisms producing the patterns that we'll see in part 1. And then briefly at the end, I'll talk about some of my ongoing and future research goals, and especially how these projects can interface with both my teaching and my outreach as an assistant professor here at the University of Michigan. This is a photo of our the University of Michigan and Peruvian research team that was on our recent expedition to the Amazon, and I'll talk a little bit about that today as well. So first, I want to make sure that everybody's on the same page when I use the term mimicry. So there are two main types of mimicry, each named for the man that proposed it, Fritz Mueller and Henry Bates, which perhaps not coincidentally, both based their work on observations they made in the Amazon during the 1800s, which in this artist's representation involves getting attacked by ferocious flocks of toucans, which I can assure you doesn't actually happen. So in the first kind of mimicry, Muellerian mimicry, multiple toxic or noxious species converge on the same warning phenotype for signaling their toxicity to their predators. Now, a great example of this is bees and vespid wasps. They're not closely related, but yet they've converged upon a similar yellow and black banded coloration, which is an honest signal of their toxicity to other organisms. So in this kind of system, it's sort of like every species is simultaneously a model and a mimic. In the other kind of mimicry, Batesian mimicry, there is a toxic species that has an honest signal, but there are also harmless species that are called mimics, which are giving a dishonest signal because although they have the exact same color pattern as the toxic models, they're completely palatable. Now, in both cases, this phenomenon results in multiple independent origins of the same trait, which is it's key to the test that we want to conduct. So now it's this latter type of mimicry that I'll be focusing on today, and specifically the Batesian mimicry of New World coral snakes, most of which are in the genus Micurus. This is one species indicated here. Now, coral snakes are highly venomous, and they're complete not only with bright warning coloration, but also with a striking behavioral warning display, involving a lot of tail waving and body flattening and thrashing while the snake hides its head. There are at least 80 species of coral snakes distributed across almost every biome in the Western Hemisphere. Their main predators are tetrachromatic birds with excellent color vision, in contrast to the coral snakes themselves, which have much more limited capacity for color discrimination. Now, although there is variation across coral snakes in warning coloration, all species have a color pattern that's some permutation of red and black banding. And most excitingly, there have been more than 150 species identified with the same warning coloration, but that appear to be quite harmless to predators. These are mostly in the family Colubridae, and they represent an unknown number of origins. So now, because I'm going to show a lot of photos in this talk that look like this, I think it's important for me to emphasize here why this color pattern is something special. So for those of you who don't spend a lot of time trying to find snakes in nature, it's easy to get the impression that this is just another typical snake color pattern. But herpetologists literally spend thousands of hours staring at leaf litter like this, trying to hone the perfect search image in order to visually resolve the snake that's sitting right here. This is a fer-de-lance, which is a kind of viper coiled up against the buttress of a tree. And so that's why when early naturalists were tromping through the Amazon, finding a bright red and black snake was something of note because they're so easy to detect compared to something like this. And this observation was even more remarkable when it was realized that actually many of those brightly colored snakes were pretty harmless to humans, and which was in stark contrast to the coral snakes that could easily kill you. So why has coral snake mimicry been controversial? Well, that has to do with the theoretical expectations of how mimicry is supposed to operate. So Alfred Russel Wallace was actually the first to write a formalization of mimicry theory in the 1860s, and he settled upon 3 logical requirements that should be true for mimicry to persist over time. And these laws, despite their age, have actually held up quite well for insects. Models and mimics are generally found in the exact same geographic area, especially in the tropics, where clines of phenotypic variation tend to be quite sharp. The mimetic phenotype is almost always more rare than a cryptic phenotype, and the mimics are almost always less abundant than the models. Now, it's important here to note that nearly all of what we know about Batesian mimicry, the theory, the observations, everything, comes from butterflies. But there are a wide variety of systems that have Batesian mimicry: frogs, hoverflies, even fish. But my favorite examples among invertebrates are organisms that mimic ants, where we see examples of convergence that are so extreme that they are very difficult to explain through any other process besides Batesian mimicry. Here you can even see this is a tree hopper with part of its thorax in a surprisingly close rendition of one of the sympatric ant species. And these kind of examples have led to a very inclusive perspective to the study of mimicry in insects, where pretty much every example of striking pattern matching to something that's toxic is pretty much considered mimicry, at least until proven otherwise. But this has not been the case for snakes. It's been a very exclusive perspective and lots of controversy that's led to a debate that's been raging for about 100 years. So many researchers actually advocate the perspective that coral snake mimicry doesn't exist at all, or if it does, it's an extremely limited phenomenon, restricted to just a handful of species in just a few places. Now, things came to a head in 1981 when Harry Green and Roy McDermott wrote a paper in Science that they actually had to title Coral Snake Mimicry, Does It Occur?, in which they argue, among other things, that for at least some places, these three rules are indeed satisfied by coral snakes and their mimics, and that this coloration is indeed conspicuous, which had been a point of major debate. They have a gorgeous figure in their paper showing variation in color pattern matching across Central America. In each of these species pairs here, the lethal coral snake is here on the left with the rounder snout, and the totally harmless mimic is here on the right with the more pointy snout. Now, although this paper convincingly showed that there are circumstances under which these rules are met, they did not fully quiet the critics, who were noting legitimate problems. And the problems are mainly with Rule 1 and 3. So lots of researchers have pointed out that there appear to be mimics outside the geographic range of the models, both north of the distributional limit of coral snakes in the United States, but also outside of the Western Hemisphere. And in fact, snakes have the only known example of an undefended mimic completely outside the range of any model species. This is the California mountain king snake, Lampropeltis sonata, whose geographic range is shown here in black, which never gets closer than about 200 kilometers to the nearest coral snake, shown here in red. And in terms of individual abundance, these colubrids mimics can sometimes outnumber venomous coral snakes by quite a lot. Now, these observations create a legitimate problem for explaining the persistence of such a conspicuous phenotype on a completely palatable animal, especially over evolutionary time. But yet, we can't ignore the striking pattern of phenotypic pattern matching that we see in snakes, like in the picture that Harry Green and Roy McDermott had in their paper and so elegantly illustrated. So the legacy of this controversy has been a whole lot of lost opportunity. Snakes have simply been left out of nearly all major reviews of mimicry over the last 20 years. There are no comparisons really being made to insect systems, and this is a shame because snakes actually represent the best and most well-studied comparison to butterfly mimicry, and there's a lot to be gained in understanding both. Now, this tension between theory and observation has led to two possible alternative explanations other than coral snake mimicry doesn't exist at all. And you can categorize these as either better tests or different rules. So maybe the mimicry theory that we've developed for insects, especially our ways of interpreting costs to predators in making an identification mistake, are simply inadequate for predicting the dynamics across the full spectrum of mimicry systems. If you have big lethal models and innate avoidance of those models by birds, which has definitely been found, those just might need modified theory. Secondly, if you're looking for correlations between models and mimics in terms of phenotypes, we know that species do not represent statistically independent data points. So first, species are not randomly distributed across space. There are continental patterns of diversity, like the latitudinal gradient, and strong spatial non-independence relative to other species, especially within the same clade. Secondly, we know that distributions of phenotypic traits often have phylogenetic signal, so you have to account for that kind of non-independence as well. This is a figure from a classic paper by Ted Garland, showing the importance of phylogenetically informed tests of trait correlations and reminding us that traditional statistics assume a star phylogeny, which is rarely a realistic assumption. So what we had was just a handful of point observations. This model mimic species pair over here seems to conform to expectations. This other one over here seems not to. But what we didn't have was a systematic unification of all the data, and that's what I decided to do. The first comprehensive statistical test of mimicry across the entire Western Hemisphere and the 80 million years that snakes have been in the New World. I used two complementary approaches to test for a correlation between models and mimics over space and over time, and I'm going to describe both approaches here and then present all of the results. So first, we phenotyped all of the New World snake species for coral snake coloration, which is best thought of as simply red and black banding. that we had a few reviews that had helped us out with this, but we also updated and standardized all of the taxonomy. We added new species that hadn't been available to the original authors and added more information about variation and coloration. Next, we constructed geographic range polygons for as many species as we could. Now, for the coral snakes, we hand constructed these ranges from published sources, mostly books. But for most of the snakes, we made these ranges based on museum data. Now, I want to highlight here that this was a major undertaking. So there's no database that you can go to and just download the geographic ranges of the snakes of the world. Even for most countries in the Americas, there's not even a book that's the reptiles of that country. And in fact, for the majority of snakes, the only information about their distribution is what's present in the collections of natural history museums around the world. And it's thanks to the huge digitization efforts of these museums to get their specimens publicly, their information for their specimens publicly available, that we were able to find usable data for almost 300,000 specimens of snakes across the New World. Then we want to do something very simple. Just simply compare the patterns of species richness. We're looking for positive correlations between models and mimics while also accounting for patterns of overall species richness. Then, for the phylogenetic approach, we augmented our phenotypic data set across all Old World species, specifically looking for this red and black banding. Then simply we reconstructed the origins of the mimetic phenotype, or at least the red and black banded phenotype, and looked at changes in rates of phenotypic evolution. But specifically, we wanted to compare the timing. When does this red and black coloration arise in colubrids relative to coral snakes? Simply asking, do coral snakes predict mimicry? And if so, how well? So what do the data look like? What you see here is an overlay of every species range polygon that then have raster counts for every latitude and longitude grid cell. These counts are then colored as a heat map, where places with red have more species and places with what should be kind of tannish have fewer. And what do we see? Well, we see a classic pattern of a latitudinal diversity gradient, where the center of snake diversity is here in the tropics. What about for coral snakes? We see a somewhat similar pattern, except it seems to be more restricted here to the western Amazon basin, so in Ecuador and Peru, and not spread across the entire Amazon. So what about mimics? So at first glance, it seems that mimics generally look pretty similar to all snakes. You find lots of mimics where you get lots of snakes, and that's pretty much it. When you run a spatial autoregressive model on this, this is just a regression that accounts for spatial autocorrelation, we find that mimetic richness is positively correlated with both total species richness and coral snake species richness. But we weren't 100% confident in these results. This is a parametric test, and we weren't sure how well it was doing at handling non-normality in the distributions of our species richness counts, because they were decidedly non-normal. We also weren't quite sure how they were handling the complex shapes of continents, because the weighting matrices for distance sort of assume a square, and that's not the shape that we had at all. So we used a permutation test. What we did is we randomized the species ID tags that are associated with all geographic ranges that were not coral snakes. Doing this, we could simulate a null expectation of how many mimics you should expect in a locality based only on species richness. Then we want to visualize the residual variation left over after you remove this effect. So we're asking, where are there too many mimics given you know that this effect occurs? And so what you're seeing here is, instead of species counts, you're looking at standardized residuals. And we're specifically looking for places where the observed value of mimics is much higher than the expected value, and that's what you see mapped. The threshold for significance is 2 standard deviations above the mean, although we did see extreme values that were almost to 5 standard deviations above the mean. And moreover, these extreme values had strong geographic signal that matched coral snake richness patterns very well. So if you remember no other slide from this talk, remember this one. Despite all the potential sources of error we had in our geographic range reconstructions, of which there were numerous sources, coral snakes pretty much explain all of the residual variation in the distribution of mimetic colubrids once you account for overall species richness. And I think that's pretty amazing. So to look at these results in another way, here in gray are the simulated expected mean values of mimics for all grid cells with the corresponding number of coral snakes. what you see here is just the correlation between coral snakes and mimetic snakes that you would expect based solely on species richness alone. But the black dots are the observed data, and what we see is a strikingly increased correlation such that there are 2 to 3 times more mimetic species than expected. And actually, the relationship is surprisingly linear with a slope of about 2, suggesting that for every additional coral snake species, the system can support 2 more mimetic species, which is way too many mimics. But what about the abundance of individuals, which is perhaps the better test? Just because there are multiple mimetic species, if they're super rare and coral snake species are super common, the abundance situation could still be that coral snakes are more common than their mimics. So remember, we have 300,000 museum records, and so we can actually test this. We can plot the ratio of mimics to models. My gray line is a little light there, sorry. But that's a reference line at 0, representing locations where models and mimics are equally common. So if you have a situation where mimics are more abundant, you should see that locality showing up on this side of the reference line. If coral snakes are more common, it'll be on that side. So what do the data actually look like? When we plot all of the grid cells, we find this. Although there are definitely places where coral snake specimens are actually more common than their mimics, and sometimes 3 to 4 times more common if you note the log2 scale, the overarching pattern is that mimics are more abundant in collections than coral snakes from the same area, sometimes to great excess. And as an independent estimate, I've also overlaid here the ratio of mimics to models that I observed on a recent trip to Peru. And you see that it's about average, but definitely mimics were more common. So I think that there's a discussion to be had about whether collections are a good proxy for real ecological abundance. But these ratios are so skewed that I think it's hard to explain these results by collection bias alone. And I think that the general pattern is a real one, such that the overabundance of mimics is actually the common state rather than the exception. So now that we've looked at spatial correlations, what about temporal ones? So what we have here, ooh, that doesn't look super great either, is a time-calibrated phylogeny of all snakes. The major clades here are marked. So remember that coral snakes are lapids, so they're going to be here, kind of at the base of this clade. And what I'm referring to as colubrids are basically these subclades up here, the dipsodines, the natrocines, and the colubrines. So we're looking for a correlation between models and mimics in time. We reconstructed color evolution across all snakes using 3 different methods. These methods were generally concordant, so I'm only going to show the results from the maximum likelihood and stochastic character mapping. So now this tree, it's not showing up well, sorry guys. It's got a number of different things marked. So first you can see these yellow circles. So snakes are primarily an old world clade that has made multiple independent arrivals in the New World. And those are each marked by these yellow circles. Then every node that we reconstructed as having the red and black banded phenotype, the descending branch is then colored by either red if it's a taxon that's in the New World, or this brown color if the taxon is in the Old World. And what do we see? We see that there is indeed a background transition rate, not surprisingly, just like any other trait. This red and black banded phenotype was present across the entire tree at a low frequency. However, those transition rates skyrocket in the New World colubrids. Now, a lot of the transitions in color phenotype were recovered at the tips of the tree. Overall, we recovered about 60 origins and about 30 losses. But those could be influenced pretty heavily by taxonomic uncertainty if they're at the tips of the tree. So the numbers that I give here for origins and losses that we found are all deeply nested transitions that are clearly robust and independent. I think that there are 2 important takeaways from this analysis. First, the only clade in which red and black banded coloration spans the transition between the Old World and the New World is in the models, the coral snakes. In every case of an origin within New World colubrids, the coloration evolved after they were sympatric with coral snakes in the New World, which is the key prediction of Batesian mimicry. But secondly, we see that it's not just once a mimic, always a mimic. There's a lot of phenotypic change, and that changes in both directions. And this is of particular note because despite a lot of trait evolution studies in insect mimicry systems, no losses have ever been recovered, at least not irrefutably. And the strength of this empirical evidence has led a number of researchers to hypothesize that mimicry is a one-way street. in which no mimics ever revert back to the cryptic phenotype. Now, there have been two reasons that have been hypothesized to explain this. So first, it's possible that the geographic ranges of insects are oddly stable, but I'm not sure that there's a whole lot of data supporting that. And perhaps what's more likely is that it's due to the genetic architecture of coloration components. So in all the Batesian mimicry systems for which we understand the genetics of the mimetic coloration, the multiple pigmentation genes that are involved in making the complete signal are all physically linked in small chromosomal regions called supergenes. And given this architecture, it may be very difficult for butterflies to quickly deconstruct the mimetic signal when in allopatry to their model. And in that case, such allopatric populations would quickly go extinct, and thus be invisible when you're making a phylogeny of extant taxa. Now, I think that this is an interesting idea, and I'm not sure we have a whole lot of evidence for this either, but what we can say for sure is that this one-way street is not what we see in snakes. So we find lots of losses, and I don't have time for all the analyses that I did, but I can say that they aren't clearly related to allopatry with coral snakes, which is a bit surprising. But bringing this back to our set of alternative explanations for why snake mimicry systems seem so different. Is it better tests or different rules that are accounting for this conflict between predicted and observed distributions of the mimetic phenotype? And I would argue that it's actually both. Some of the conflict is explained through better tests, and some of the conflict is even worse than historically suggested. So coral snakes explain way too much of the variation in the distribution of the red and black banded phenotype to argue that coral snake mimicry doesn't exist or even is a very restricted phenomenon. And most importantly, if you're looking for correlations between models and mimics, you can't treat each species as an independent data point. Although I would say, at least superficially, this non-independence seems to be more important in snakes than it is in insects. But despite the overwhelming support that we found for Batesian mimicry, we also found or confirmed patterns that you just don't see in insect mimicry systems, suggesting that the rules may be different too. And specifically, these requirements for an increased abundance of models or for sympatry between models and mimics seem to just not be applicable to snakes. And that the evolutionary lability of the red and black banded phenotype seems to be pervasive and extreme, and includes numerous losses. So I would argue that if you really want to understand mimicry, snakes provide a very important comparison to insects, and especially to butterfly mimicry, and that those comparisons are probably of the greatest importance going forward to figure out why these discrepancies exist. But I have oversimplified everything that I've shown you so far, and there's one key factor in particular that I left out. And that's that snake mimicry systems, just like insect mimicry systems, have spectacular color polymorphism, in which multiple, very different color types coexist sympatrically within single populations of single species of both models and in mimics. So the species shown here is one of my main study species. This is the western ground snake, Sonora semi-annulata. And under a single rock, you can find 4 different color phenotypes. A mimetic form of red and black bands, ones with just the black bands, ones with just the red, and ones that seem to have neither color component. So in conducting our global phenotyping across all snakes, we absolutely paid attention to this color variation, in addition to simply noting whether one of the morphs had this red and black banded phenotype. And in doing so, we determined that at least 1/3 of all mimics and 1/5 of all coral snakes have a major color polymorphism for either the red or the black banding. And that's almost assuredly an underestimate, given how this kind of polymorphism is reported in the literature. So that brings us to our second major theme, in which we use this color variation as an opportunity that allows us to follow the mimetic phenotype over time and space in a focused way, looking at a single clade in depth. And specifically, there are 2 goals here. First, focusing on the ecological interactions between the models, the mimics, and the predators. What is the relationship between predator and prey? Do predators avoid the red and black phenotype as predicted by mimicry theory? And secondly, focusing on a single clade allows us to investigate the genetics of pigmentation. Many studies have suggested that biases in the direction and frequency of phenotypic change may be due to the genetic mechanisms that are underlying trait development. But how does snake mimicry compare to butterfly mimicry? Do they have supergenes too? And as an overarching idea over all of this, what is the role of the color polymorphism? So the clade that I looked at in depth is this tribe Sonorini. They're a clade of small secretive snakes in North and Central America. There are about 25 species. They have various levels of color polymorphism and of mimicry, although the extent of both is pretty unknown before I started working on them. But the reason I chose them is because their northern distribution and the fact that they specialize on eating insects means that they reach abundances that are more typical of lizards than of snakes. That means they're relatively easy to catch in very large numbers, which is not true for most of the tropical species. So the original heat map that I showed you of coral snake species richness made it seem that all the excitement was in South America. And while that's true, coral snakes in North America are still quite interesting, with lots of color variation among species and complicated patterns of geographic range overlap, both with other coral snake species and with their mimics. So this is all of the species of Sonorini that are also mapped. I've simply randomly colored ranges by species here with transparency so you can visualize some of the complexity. Now the Sonorini species do have a similar distribution to coral snakes, although there are more of them. But I mostly focused on this top species here in yellow, which has areas of both allopatry and sympatry to coral snakes. And they overlap at the part of the ranges of at least 3 different species of coral snakes here. So in order to test hypotheses about selection and the genetic control of coloration, I compiled 2 different data sets, a phenotypic data set and a genetic data set. Now, sonora are mainly captured through rock turn surveys, as I'm demonstrating here, where you flip over a lot of rocks and take snakes out from underneath. From our work, we estimate you have to flip about 1,000 rocks for every sonora that you catch. So we augmented our field data collection with some museum specimens as well, but we ended up with a total data set of almost 3,000 individuals. We also collected 3 different genetic data sets. Today I'm only going to be showing the data from the AFLP analyses, but you should know that the other data sets do exist as well. So first, looking at the distribution of morphs across space. Here you're going to see data for 180 counties in the United States, and the height of each of the bars is proportional to the number of individuals in that county with that phenotype. So first we see that the mimetic phenotype is distributed all across the continental United States within the range of the species. Even up in Idaho, where actually the original specimen that was found there in 1923 was actually misidentified as a coral snake. However, by far the most common morphs seems to be this uniform brown morph and the one with only red. More importantly, coloration seems to be completely decoupled from any environmental variable that we can find. So it doesn't matter whether you're looking at the presence or absence of morphs, the proportion of morphs, the proportions of ones that just have black, ones that just have red, and it doesn't matter whether you're looking at latitude, longitude, sympatry with models, anything we can think of, we don't see any correlation. So this immediately begs the question, is this phenotypic variation neutral or is it driven by selection? So the way that we tested that was to simply compare genetic and phenotypic variation for all the populations for which we had at least 20 contemporary samples. We needed tissue samples. So for example, if you're looking for a signature of either genetic bottlenecks or drift, you can imagine a situation where if a population has gone through a bottleneck, you get reduced genetic variation and you should have lost phenotypic variation as well. And so you should get a positive correlation between these 2 variables. What do we see? Nope, no correlation there either. So what about genetic distance versus phenotypic distance? So you can imagine if populations are sharing a lot of migrants, they're going to be pretty similar genetically, and they should also be pretty similar phenotypically. Do we see a positive relationship here? No, we don't. In fact, it's only when you compare FSTs, where we found very little genetic variation among populations. The average is about 0.06, but phenotypically, populations were quite different. And this kind of pattern is the classic signature of phenotypic traits that are under selection. But what kind of selection? So the way that we tested that was in 2 different approaches. First, we compared age classes within a single generation. Do you see different frequencies of the color morphs in juveniles versus adults? So here what you can see is for every gray bar, that's a set of juveniles, and for every black bar, that's your adults. And we see that there are quite different phenotypic frequencies across the different color classes, which is another classic signature of selection. But what about it changes in morph frequency over many generations? Anytime you have a color polymorphism that seems to be maintained by selection, you probably should be thinking about negative frequency dependent selection. Basically, an increase in selective advantage when the morph is rare. This is a classic mechanism for maintaining polymorphism within populations. So the way to look for that is to simply For every population where you have a long capture history from your museum specimens, use multinomial logistic regressions to basically look at the change in morph frequencies over time. And what do we find? So yes, we found very convincing evidence of negative frequency-dependent selection. So each of these lines is a different one of the morphs, and that the probability is simply a proxy for the frequency, the real frequency of the morph within the populations. And you see very clear cycling of morph frequencies over time, with the morph that was the most rare at the beginning of the time window becoming one of the more common ones by the end. So we didn't just find this in a single population, but we found it in every population for which we had a long enough time sampling interval to actually perform this test. So the reason why this is important is because it makes morphs hard to lose. So even in allopatry to the models, if a morph is under strong selection, positive selection, when it's rare, it can be very hard to eliminate from the population entirely. Now this is an exciting study because it's the first direct assessment of selection on color pattern in a snake mimicry system. And I want to be clear that what I mean is that it's not using clay replicas. So rather than focusing on how predators interpret visual stimuli, looking at their attack rate on a clay replica you put out on the ground, what you're actually looking at is the real response of prey populations over time to predation, which is pretty exciting. So the one other thing that you can do with this data set is to examine the genetic architecture of the mimetic signal. So although breeding studies in the laboratory are always the gold standard because of the kind of control you have within a laboratory setting, not all taxa are amenable to large-scale breeding studies to address things like genetic control over coloration. And Sonora is definitely one of those species that's not super amenable to that. But with a whole lot of population level data on the observed morph frequencies and some basic population genetics, you can make some inferences about the likelihood of some very simple models of genetic inheritance. And these are the simplest models that could conceivably produce the 4 phenotypes that you see here. So a one locus model where you have 3 alleles, one for red, one for black, one for null. They're co-dominant, so if you have an R and a B, then you're mimetic. If you have 2 nulls, you're uniform, and so on and so forth. It could be that red and black are actually 2 different loci, in which case you have 4 alleles, a red, a null for the red, a black, and a null for the black. Or it could be that these 2 loci are quite linked. And you can imagine a situation under total leakage disequilibrium. You would find populations that have all mimetic and all uniform, but not any of the other 2 types. So you can simply calculate these expected phenotypic frequencies under Hardy-Weinberg for these different models and do a quick comparison of the fit of the models to the observed data and generate AIC scores. So which model is more likely? So what I'm going to show you here are the delta AIC scores for each of the 40 populations that we had with enough data to actually perform this test. So here's the reference line, here it's 0. Any population that falls along the reference line would be pretty uninformative. It doesn't support one model or the other. If it's above the line, in this plot, it's going to support a 2 locus model over a 1 locus. And if it's above the line in this one, it's going to support the unlinked model versus the tightly linked model, or supergene model. So what do the data look like? Well, there are lots of populations that are actually completely uninformative. Mostly these are populations along the Great Plains, if you think back to the 3D maps, that are that have a high frequency of the uniform morph. And that really doesn't tell you a whole lot about the control of the red and the black. But for the populations that did have high information content, we see very strong support for red and black being controlled by separate loci, and support that those loci are not linked. And in fact, the model with the very strong or supergene linkage was actually a worse fit than the 1 locus model. And this is interesting because finding color components maintained on separate chromosomes has only ever been described for Mullerian mimics. And in all the Batesian mimics that have been looked at, all we ever find are supergenes. So it's possible that Sonora are an anomaly, right? They represent an atypical breakdown of genetic linkage. But if that's not true, if these are an accurate representation of the coloration genetics across snakes more widely, then there are 2 important ramifications of this finding. First, because the mimetic signal can be broken down through simple recombination, you should expect the trait to evolve very quickly. And you should be able to predict what kinds of morphs that you see that are as components of the total signal. And both of these may work together to be part of the explanation for the extreme lability that we see in mimetic phenotype across an entire phylogeny. So bringing it back to some of the spatial analyses that we did. Instead of looking at just the residual variation of mimics, this is now looking at the mimics that are actually polymorphic. Where do you see too much color polymorphism given total species richness? And we actually find that polymorphism seems to be too high, both in places with high coral snake species richness and in places with the lowest coral snake species richness. And to me, this suggests that there's a mixture of processes. And while it's not a perfect correlation, it seems that the cryptic morphs seem to be much more common in allopatry. And so this ends up being a key trait. It's not just, does polymorphism exist, but what is the actual nature of the polymorphism, which ends up being critical for understanding how phenotypes are interacting, especially from the perspective of the predator. But I think the overall message here is that this color polymorphism appears to be a key step in both the origin, loss, and maybe even maintenance of mimicry. So my hope is that everyone takes away from this talk a somewhat new view of mimicry, moving away from the oversimplified, easy-to-explain version of mimicry, as illustrated here by two species of frogs, in which there's a single mimetic species that perfectly imitates the color pattern of a single model species, and that we instead embrace the dynamic and complex reality that we're just beginning to fully appreciate, where single species can actually have exceptional diversity of quickly evolving phenotypes. And this represents an exciting opportunity for understanding how evolutionary convergence operates over time and space. So where do we go from here? So I have several projects for which I've been collecting preliminary data and that I currently envision as both feasible and fundable moving forward. They're a good mix of field, lab, museum work that are all geared towards the goal of better understanding the mechanisms and dynamics behind novel phenotypes. Now, I only think I have time to briefly outline one of these projects as sort of a representative of how my work promotes educational and collaborative opportunities for students at Michigan and beyond. So bringing it back for a moment to Mueller and Bates. So some of you in the audience may already have recognized that all of the coral snake mimicry problems, whether it's the abundance problems or the distributional mismatch between models and mimics, or the curious lack of genetic linkage, all of these concerns suddenly disappear if the system isn't actually Batesian, it's Muellerian. If all of the species are toxic, then it no longer matters which species are allopatric to others or what the abundances of each species are. Reinforcement of predator avoidance would then be everywhere that the snakes were. So this is not a new idea, but it's certainly not been well tested. I want to make it clear that no New World colubrid snakes have the same kind of advanced venom delivery system that you see in coral snakes. They don't have front fangs, they don't have muscular control over their venom glands, and all of those kinds of adaptations that can clearly kill predators. But a surprisingly large proportion of mimics do have a system for incapacitating their prey. So they have enlarged teeth, they're just in the rear of their mouth, and those teeth are connected to specialized glands that sit in the same place as real venom glands on elapids and on vipers, and they're actually hooked up to the teeth. Now, the components of these glandular secretions have never been really well examined systematically, and certainly not across hypothesized transitions to a venomous state. But the question begs to be asked, how toxic actually are the mimics? So before we actually formalin preserve specimens for collection, we've been removing these glands, the Duvernois glands. This is a picture of Joanna Larson doing one of these gland removals on a coral snake mimic. You simply go in through the mouth, in between the teeth and the cheek, and pull out the gland, and actually the specimens look great afterwards, and you can't even tell what we've done. So now we're ready to analyze the RNA in these glands to examine transcript differences across species, and especially across evolutionary transitions. And this will be the first such broad-scale assay in colubrids. So linking venom components to tooth evolution, so even without getting venom glands from every single species of New World snake, we can take advantage of the huge collection of preserved specimens at the UMMZ from the perspective of skeletal changes. So an upcoming senior thesis student, Aaron Westeen, who's currently a junior, is using a combination of micro-CT scanning and geometric morphometrics to phylogenetically model tooth evolution across New World colubrids. And here you can see one of her scans. This is the front of the snake here. This is the hinge of the jaw here. And I've false colored the enlarged maxillary teeth here in red, so you can see you can see how big they are relative to the rest of the teeth in the mouth. And so you can ask a bunch of questions here. How many of the teeth in the rear of the mouth are enlarged? How enlarged are they relative to the others? Is there grooving on the teeth, which is the conduit for the venom coming down out of the gland? What's the curvature like? And most excitingly, are these transitions correlated with changes to the red, black, and banded phenotype? Nobody's ever approached this question quantitatively. And I think that if you want to study something like cranial morphology across snakes, University of Michigan is an ideal place to be because it's the largest university-affiliated collection of herps in the world. So although University of Michigan is clearly a great home for me, I think it's also important to think about giving back. What opportunities do I provide? So you've seen some demonstration of some of the student opportunities that are available through my research. And also because this work is across Latin America, there's a lot of opportunity for international collaboration and outreach. But one of the most exciting opportunities for uniting research and teaching is my vision for adding more field-based courses to our catalog. So in graduate school, I was a teaching assistant for 13 courses, many of them with a strong field component. And then as a postdoc, I was a primary instructor for 2 field courses, one of them the flagship course on vertebrate natural history at UC Berkeley, and another was a destination field course to Kenya. And I think that my experience teaching field courses has led me to really appreciate the incredible capacity for field courses to inspire students both about doing their own science and also for drawing in new majors to EEB. And I think that there's a great opportunity for more of that here that I would be really excited to lead. So with the few minutes that I have left, I just want to make some important acknowledgments to people who helped make this work a reality. So first to Christian Cox. So he was a first-year PhD student when I was finishing my PhD. And we found out about each other when we started requesting the same tissues from the same museums, because we both realized the exciting capacity that Sonora had for answering questions about mimicry and polymorphism. And so instead of getting territorial, we combined forces and worked together, and it's ended up being a fantastic collaboration with each of us bringing very different strengths to the table. My postdoc advisor, Jimmy McGuire, hosted me at Berkeley and helped add a really important Southeast Asian herpetofauna view to some of our trait evolution studies. And also, when you're struggling through the difficulties of ancestral state reconstruction, it's really helpful to have a macroevolutionary modeler sitting at your kitchen table. But I also want to say, highlight some work by 2 of our EEB graduate students, Pascal Teitel, who was instrumental for the construction of the range maps for 1,000 New World snake species, and also Iris Holmes, who contributed to some of the genetic analyses that went into my work. And especially thanks to the EEB department as a whole. You guys have all been incredibly supportive, and I thank you so much for coming today. I think I have a couple minutes for questions. No questions. So the question was about if the color polymorphism might be for evading predators. So I would say probably the prediction in that situation is that you shouldn't have a conspicuous polymorphism. So you could have all different kinds of cryptic phenotypes. But the problem is once you get one that kind of looks like a stop sign, but is perfectly palatable to predators. And the idea is that once they eat one, they learn that that's a really great thing to eat, and then they can very easily specialize their search image and take them all out of the population. So I would say probably the best predictor is how conspicuous the morphs are. Yep. So I would probably, for some butterfly groups, I think we do know geographic ranges pretty well. But I think if you wanted to know, like, where are the ranges of all the butterflies that are in the Amazon, if the data don't exist for snakes, They don't exist for butterflies. And so I think it's a really exciting test. And I think with more investment in digitizing our invertebrate collections as a natural history community, yeah, I think that that's a really important test to conduct. And I'm really excited to see what that looks like compared to the results that I found in snakes. So within, oh, definitely, they're totally, you can get all four morphs within a single clutch. Yeah, absolutely. No reproductive isolation that we can find by morph. So we tested that, and I haven't analyzed the data yet. So some of you might remember Kinsey Brock. She was a master's student in SNRE. And so she had a summer here before she went on to her PhD program. And so we had a giant red-tailed hawk that was mounted that we swooped in on the snakes that we had in the lab to look at escape behavior. And she videotaped it all. And no one's gone through all the video yet. But yeah, we were interested in that because there are definitely some classic papers on correlational selection between color pattern and behavior. And so I think that's a really great question. So that's a great question too, and I would say we don't have a whole lot of data on exactly which birds are eating snakes. So predation events are pretty difficult to see in the wild, and so most of the data we have, at least for raptors, comes from a place up on the Snake River where they have a long-term raptor study, and they go and check all the nests, and there are little bits of things that get flung out of the nest, and they pick it all up and try to identify it. And so we know we have some information about some of the birds that eat snakes, but mostly we kind of wave our hands. So even robins, for example, are known to eat snakes. So sometimes it's not always the birds that we're expecting, and to try to get a really good handle on which are providing the majority of the selection pressure, yeah, we don't know, but that's a good question too. Mm-hmm. So it depends how strong you think the selection should be. I mean, if you're a population geneticist and you think something that's that conspicuous and that edible should be taken out pretty quickly by selection within a few generations, the mismatch between the coral snakes and the mimics, even if it's been a retraction or an expansion, was several thousand years ago. And so you would expect it to be gone. But I would argue that, yeah, it's hanging on, yeah. And that needs to be explained, an evolutionary lag. Sure. So at least the Pacific route is a known migratory route for birds. I think the fact that you get them going up pretty high into the Great Plains, I'd love an ornithologist's perspective on migration routes here, but I've heard that that's a little controversial about whether that's actually a migration route or whether birds mostly follow water. So you could absolutely test, if you knew bird migration routes, you could actually test very, very clearly whether or not those overlap. One test that was conducted by one of my lab mates at Santa Cruz was to actually compare the response of predators to clay models within the Sierra Nevada mountains and within the Coast Range. And those have different proportions of resident versus migratory birds, and he found no difference. So, in attack rates. And so it's hard to say whether the residents are actually behaving in a different way than the migrants, but yes, the working hypothesis would be that migration could explain at least some of the distributional mismatch.
Okay, maybe one more question.
Okay. Okay, go ahead.
I have no idea how uniform the toxic butterflies are in their color pattern, but it strikes me that coral snakes at least are really consistent from like Florida all the way down to the Amazon with red and black. So kind of related to that, could it just be that these birds that are ranging over large areas learn that red and black snake is bad, kind of no matter where they are, and just like carry that learning wherever they go?
Yeah, so I didn't show pictures of the coral snake color polymorphism, but it's rampant. So we get, because actually the species that are in the US are pretty conserved, we get this idea that yeah, it's pretty much always banding, and we have little rhymes about the order of the bands, telling you whether or not it's a safe snake to touch. But no, there are coral snakes that are completely red. There are coral snakes that are completely black. There are coral snakes that are black and white. They're usually some permutation, but they might not have bands. They could be all black. They could be all red. And so, but yes, I mean, a detailed analysis of exactly how many are tricolor versus bicolor versus unicolor might be able to tease some of that apart, but you start to lose power when you start dividing your 80 coral snakes up by color pattern. So we didn't do it here, but yeah, I think that's an exciting future target.
Okay, let's try Allison one more time.
Thanks, guys.